Yeah, thanks very much for the introduction, Borja. When Borja first approached me and asked if I wanted to give this talk, I thought it sounded like a great idea to review hyperpolarized NMR for biological systems. But then when I started putting it together, I very quickly regretted that decision because it's really an enormous field of research. And it's just about impossible to cover it in any, well, certainly in like a sort of a 30-minute talk, it's impossible to cover it in any real detail. And more than that, it's just a huge field of research, and I'm certainly not an expert in, well, most of it, to be honest. But the idea is that for the first half of the talk, I'll just give a broad overview of this topic, and then for the second half, I'll get a bit more in detail about what I'm working on here in Barcelona and how we're using hyperpolarized NMR. Okay, so I'll begin. So there's this famous quote in NMR. We said there's 3 problems to overcome for NMR: sensitivity, sensitivity, and sensitivity. And I actually don't know where this quote comes from. If anybody knows the origin, please do let me know afterwards. As far as I'm aware, it's been around for years. Well, okay, so here we are. This is what I have here is a schematic of an NMR experiment. So the idea is you have your nuclear spin system in a magnetic field, and you have a coupling between the spins and the magnetic field, and that gives you some level of thermal polarization. And then you would excite the spins and detect them. And the hyperpolarization is really concerned with this first step. It's about increasing the polarization beyond what you would get by just letting spins equilibrate in a magnetic field. Okay, so I guess we're all quite familiar with this sort of picture where we have some chemical sample, and if there's no magnetic field, the nuclear spins are all disoriented. But if we introduce a magnetic field around the sample, then they can line up with or against the field, and there is some slight net bias to align along the field. And the polarizations we can reach by employing the highest field magnets available are of the order of about 10 to the minus 5. Up to 10 to the minus 4 for protons in field strengths of above about 20 Tesla. Okay, nevertheless, that's still good enough to do NMR. So if we have an NMR sample, we can collect like a thermal equilibrium spectrum. So hyperpolarization, the idea is that you can, if you're hyperpolarizing the spins as they flow through this hypothetical device, what happens is you're selectively populating one or more nuclear spin states, and the polarization increases to, in principle, 1 or 100%. And then what you would detect after the hyperpolarization process is dramatically enhanced NMR signals. That might be one or many of the signals in the spectrum. Okay, so, well, when we talk about polarization, this is a simple formula for an ensemble of uncoupled spins. So we're simply looking at the difference in state populations divided by the sum of all state populations. And we can write this expression for, which is an approximation of the thermal equilibrium polarization you can get. And I think the important factors to note there are that, hold on one moment, let me swap my pointer to okay, hopefully this is a bit clearer. So the important points to note are that the polarization at thermal equilibrium is approximately linear with magnetic field strength and inverse linear with temperature. So here what I've plotted, just to give a picture of that, is the polarizations of 3 different spin half particles at a magnetic field strength of 11.7 Tesla, which would correspond to what we'd more colloquially refer to as 500 megahertz proton resonance frequency at different temperatures. So, well, essentially we see that as we decrease the temperature, the polarization goes up and it saturates at 1. And you can get to, we call this unity polarization. You can do that quite readily for electrons down at achievable temperatures of around 1 Kelvin. But for nuclear spins, that's to get to unity polarization just by cooling the sample is very difficult because you need expensive equipment to get to these millikelvin temperature ranges. Okay, and then here I just overview some key applications of hyperpolarized NMR. So in low field and portable NMR devices where you don't have much or any natural polarization. So if you're working at low field, you just don't, your thermal equilibrium polarization is very low. You might need hyperpolarization. Another case is in microfluidic devices or porous media where your sample size is limited. A similar case is where you're working with a limited concentration of sample. So a good example of that is in biomedicine, or if you're trying to study a protein, for example. And finally, I include this, which is mechanistic studies with hyperpolarization, where some hyperpolarization methods will deliver signal enhancements, but it's very specific to the mechanism through which that hyperpolarization is delivered. So you can extract information about a process. This isn't really relevant to today's talk, so I think we'll just gloss over that. Okay, so here is a simplified hierarchical diagram in an attempt to map out the whole field of hyperpolarization and give you a different way of thinking about it. So what we've got down here on the right-hand side are some common hyperpolarization techniques. And over on this left-hand side, we have the source of polarization, which is sort of external to the system. So that could be circularly polarized photons, the general concept of Boltzmann statistics, or for KIDNIP, which is chemically induced dynamic nuclear polarization. It's a bit of a special case and sort of hard to define exactly where the polarization comes from in that case. So we'll pass over that for now, and we'll just look at these two broad cases. So we can zoom in there and see and sort of run through this diagram. So we're saying that we have some external source of polarization, which is going to deliver hyperpolarization to a source that's internal to the system. And that internal source couples to the nuclear spins. So there's some sort of coupling between the electrons and the nuclear spins. So that could be well, it's essentially in all cases a hyperfine interaction, whether that's dipolar couplings or the Fermi contact interaction. We then have some sort of mechanism through which the polarization is transferred from the electrons to the nuclear spins. And then, well, and finally, the technique that corresponds to. So this is spin exchange optical pumping. And I don't want to go into the details of these techniques too much. The point that I want to highlight here is that in just about all hyperpolarization techniques, the polarization comes from either Boltzmann statistics or circularly polarized photons. And when we say Boltzmann statistics, what we mean is there's some degree of freedom in the system that you can polarize to a high degree. So that might be electron spins themselves. So you can have electrons in a sample, and we've seen that just by cooling them down in a high magnetic field, the electrons can reach unity polarization. And then you can transfer the polarization from those electrons to the nuclear spins. So those methods are broadly, there are many techniques to do that, and they're all dynamic nuclear polarization techniques. Another really neat example is that rotational energy levels, the energy differences involved there can be quite large, certainly much larger than for nuclear spin states. So if you can generate rotational polarization and transfer that to nuclear spins, well, then that's another method to hyperpolarize them. And there are a few techniques there. I think the standout one is para-hydrogen-induced polarization. We also have a fun case down here where the source of polarization is simply off the nuclear spins themselves. So, well, there's no concept of coupling there or polarization transfer. So these techniques are brute force and statistical polarization. Brute force being simply cooling down the spin system to very low temperatures and just letting it become polarized. Okay, so with that sort of diagram in mind, I'll just highlight these. So we have, I'm going to be talking about spin exchange optical pumping, para-hydrogen-induced polarization, and a couple of DNP techniques. I think these are the sort of most relevant for biological systems, the most widely applied, let's say. Okay, so the first technique we'll look at is spin exchange optical pumping. So with this technique, all right, we can go through exactly what's going on and how it works, and then I'll talk a bit about applications. So you have the setup here where you've got a rubidium vapor cell rubidium is shown in red, and that's floating around with xenon atoms. And you irradiate the rubidium with circularly polarized photons. So circularly polarized is indicated by sigma plus here. And it means that those photons carry angular momentum. And so when they interact with the rubidium atoms, they can excite an electron, but they can only excite it with a corresponding electron spin flip. So the electrons can be excited in this sense, going from, let's say, spin down to spin up, but not the other way. And then you have relaxation. So essentially, after doing this for some time, you pump the electrons up here, and then they relax down to here, and you keep doing that, and you accumulate electron population in this state. So that's how the circularly polarized photons can lead to electron spin polarization, electron hyperpolarization in this case. And then that is transferred to the xenon atoms through the Fermi contact interaction in 3-body collisions. So these atoms are floating around in the vapor cell, and they bump into each other. And you have these spin exchange interactions. So the xenon becomes polarized and flows out of the cell. And it can be collected in liquid nitrogen. It'll simply freeze out as a snow or used as a gas. Well, I think this hyperpolarization technique is beautiful because this equipment's not very expensive and you can reach very high xenon polarizations. But the really big limitation is that it's only relevant to noble gas atoms, because any other molecules would simply be ripped apart in that sort of environment, a hot vapor cell with rubidium atoms floating around. So, but nevertheless, if we can polarize helium-3 and xenon in this way, it can be used for biomedical imaging. So this is used by you breathe in the gas, and an image is taken of the lungs. And what I also show down here is that the xenon can pass through this barrier in the lungs into the blood, and the xenon has a different chemical shift based on its environment. So if it's in red blood cells, you can detect a difference. So you can start to do imaging of this blood region around the lungs. And that works for xenon because it's highly soluble, well, not highly soluble, but 10 times more soluble in blood than helium. Okay, another technique is magic angle spinning dynamic nuclear polarization. So in this technique, you take a sample and you, well, here we go, let's run through the equipment. So you have a gyroton to generate microwaves and an NMR magnet where you have a sample in a rotor and you're spinning it at the magic angle. And if the sample is frozen at low temperature with some free radicals in there, a source of electrons, the electrons can be polarized by microwave irradiation. The electrons are polarized by the low temperature conditions, and the microwaves allow you to transfer the polarization from the electrons to nuclear spins. So, there are some great advantages to this technique. I think the fact that it's quite non-specific is a big advantage. So you can essentially freeze more or less anything with a free radical and polarize it in this way. And it's used at the moment for structure determination, but it has some drawbacks in that, well, quite clearly the equipment requirements are relatively expensive. But nevertheless, it's used for, so people have studied, people have done in-cell NMR, where they've frozen eukaryotic cells and spun them and applied microwaves to generate these enhanced spectra. I think in this case, they're studying ubiquitin. And then down here, we have another example where the structure of RNA is being looked into, and you can introduce paramagnetic metal ions. And there's a neat experiment where they look at two cases. Either this acts as a relaxation agent, so some peaks in the spectra are diminished, and you can determine what's in close proximity to the paramagnetic metal ion based on which peaks are decreased in intensity. Or you can use this to do the DNP process itself. This manganese acts as the source of electron spins. Okay, and then the other dynamic nuclear polarization technique is dissolution dynamic nuclear polarization, quite similar. So In this case, you also have a frozen sample. You freeze your sample with radicals embedded in the sample. So here's the example of a radical, and you're transferring the electron polarization to nuclear spins nearby. But in this case, you're not spinning the sample because it's not going to be detected in situ. What you do is you use a pressurized hot solvent to dissolve the sample and flush it out into another measurement device, normally an NMR or MRI magnet, and you measure it over here. Well, this is really, you can reach high polarization levels on many molecules. And just like magic angle spinning DMP, it's quite non-specific. You can polarize lots of things, but it suffers from many of the same drawbacks. The polarization build-up times being a notable one here. It takes, well, on the order of about 30 minutes or so to polarize a sample in this way. Well, this is because you can enhance NMR signals so much in this way. Some neat experiments have been shown where you can use 2D NMR sequences. So this is like a conventional 2D cozy experiment. And you can perform that in what we call an ultra-fast way, where you use gradients to spatially encode parts of the sample. And you can acquire a spectrum like this. Well, in this example, it's in 200 milliseconds to acquire a similar cozy spectrum, where here it took 20 minutes. In other dissolution DNP experiments, you can hyperpolarize water. So you hyperpolarize water in the polarizer here, and then you flush that out into an NMR magnet, and that's mixed with a protein sample. Hope everybody can see that. That's mixed with a protein sample, and the water can exchange with any available exchanging sites on the protein, and then those residues become the signals are enhanced there. And so what's shown here by the different colors is which residues show the highest signal enhancement. So you can get an idea of which residues are exposed to the solvent versus ones that are perhaps coiled up and hidden in the hydrophobic core of the protein. And so this is like a 2D NMR spectrum, proton nitrogen showing signal enhancements in specific residues in red. In another example, this is done with an intrinsically disordered protein, and Borja showed me this yesterday, that in this case, because the protein's disordered and there's a lot more flexibility, you see that the signal-enhanced carbon-nitrogen 2D map is almost identical to one taken at thermal equilibrium, albeit with larger signals. But that's because almost all of the residues are in exchange with the water solvent. And I think I should clarify, I think I'm right in saying this is performed in D2O with just a very small amount of H2O. So that the lifetime of the polarization is longer than just a few seconds. Okay, and now I think I can say probably perhaps the most well-known use of hyperpolarization in NMR is for metabolic imaging. And this is where dissolution DNP has played a really big role. So this is a thermal equilibrium proton MRI image. I guess everybody's quite familiar with this sort of image where you're looking at water or fat in the body. And you can do that because even though MRI is a very low sensitivity technique, there's so much water in the body. There's so many water molecules that the concentration's high, and you can collect these sort of images. But going further than that, if you could hyperpolarize metabolites, which are at much lower concentration in the body, if you're able to hyperpolarize them and increase the size of the MRI signals, then it allows you to do metabolic imaging where you're tracking the metabolism from one metabolite into another, or more steps in a metabolic pathway. And this is being done. This is actually done in humans at the moment. This is an exciting technique, and this for the rest of this talk I'll be focusing on applications of hyperpolarization in metabolic imaging. Okay, and then, well, here I've labeled these with the carbon-13, and the question is why carbon-13 here? well, there's three reasons why this is done with carbon-13 and not proton. That's that there's essentially no background to these images. So you don't have this large water background. You have a large chemical shift dispersion with carbon-13, so you can easily see the difference in the metabolites in the spectra. And then the really standout reason is that the T1 of the carbon-13 is typically longer than that of a proton. So the T1s you can expect for these sort of molecules, or put another way, the lifetime of this tracer in the body is of the order of 30 or 60 seconds. Okay, and then, well, this is an explanation as to why pyruvate to lactate conversion is the current gold standard for this sort of in vivo metabolic imaging. There's something called the Warburg effect, where healthy cells uptake glucose, and that's converted into pyruvate, and then that can be converted to lactate or enter the Krebs cycle, the citric acid cycle. But it's been shown that cancerous cells have an elevated rate of glucose uptake, and the pyruvate preferentially is converted into lactate. So what we have here is, this is a thermal equilibrium proton MRI image showing a prostate tumor in part of the body. But if you then inject hyperpolarized pyruvate and observe the lactate to pyruvate ratio, you see a corresponding tumor area here, but also one over here in this part of the image that you didn't see in the proton image. So what we can say in general is that by observing changes in tissue metabolism, we can see disease progression or treatment response before structural changes appear. So it can be that the metabolic flux is changed in a disease part of the body before there's any significant structural change that you would be able to see with conventional MRI. And here I show another example of a different metabolite that can be used in vivo. This is fumarate, and it can be used as a marker for cell necrosis. So the idea here is that the cell membrane has this dicarboxylic acid transporter protein, and the rate at which fumarate can pass through here and enter cells is very slow with respect to the relaxation time of the hyperpolarization. So if you have a healthy cell, the hyperpolarized fumarate is essentially stuck outside, and it doesn't get into the cell before it relaxes and the signal is lost. But if the cell is undergoing necrosis and the cell membrane is ruptured, these enzymes, fumarase, can enter the extracellular space and convert the fumarate into malate. So hyperpolarized fumarate is a marker in vivo for cell necrosis. And I think here we have a nice example of an infarct heart and a control mouse with a healthy heart. And we can look at the hyperpolarized fumarate map and the hyperpolarized malate map. And we essentially see that for a healthy mouse, we don't have malate forming, whereas we do when the heart was infarct, because some of the cells died and this enzyme was allowed to escape. Okay, so as I've said, dynamic nuclear polarization is the current gold standard for producing these hyperpolarized molecules. Two clear reasons for that: you can polarize many things, and the polarization levels you can reach are typically above 20%. But there are drawbacks to that technique that we've gone over. And so an alternative method is emerging called parahydrogen-induced polarization. And with this method, you can create hydrogen gas in, well, let's call it a hyperpolarized state, parahydrogen. It's probably not quite correct to call it hyperpolarized because it carries no net magnetization, parahydrogen, but you know, for the sake of this talk, let's say you can produce this hyperpolarized form of hydrogen gas. Well, that's done very easily by passing the gas through a low temperature region at about 30 Kelvin. Okay, and then the hydrogen can be reacted with molecules to hyperpolarize them. So if we look at this example here, the hydrogen is brought into this molecule, it reacts across this triple bond. The polarization is then transferred from the hydrogens to a carbon-13. And finally, this part of the molecule is cleaved off. This is all done in an organic solvent. And this part of the molecule is cleaved off and remains in the organic solvent along with the hydrogenation catalyst. And pure pyruvate is migrates into the aqueous phase. So this is a way of producing pure hyperpolarized pyruvate through parahydrogen. So there's some drawbacks to that. The polarization levels are typically lower. It's highly chemically specific. I mean, you can see clearly that you need some part of the molecule that can be hydrogenated, and that significantly limits what you can polarize. This is what we call sidearm hydrogenation. This was developed in Torino back in 2014 and 15. And it's a really nice example of overcoming that problem of the high chemical specificity. But nevertheless, these routes have to be designed for each molecule that you want to polarize. And the solutions are contaminated. You need some way of purifying the molecule from the reaction solution. But it comes with some really big advantages. The turnover rate for these experiments is just minutes because you're just reacting. It's just a chemical reaction. And the equipment is simple and inexpensive. Okay, here's a little dive into what's actually going on there. So I said that we can hyperpolarize that hydrogen gas by simply cooling it down. So I'll explain how that works. So the hydrogen molecule behaves as a quantum rotor. You have two spin half particles, and the rotational states, I say they're coupled to the nuclear spin states by the Pauli exclusion principle. So you have a system of two coupled fermions, and the overall wave function must remain anti-symmetric with respect to particle exchange. So what it means is that if the nuclear spins are in a symmetric state, if you can exchange the two nuclear spins and the molecule looks the same, then the molecule must be in an anti-symmetric rotational state. So over here, we have a diagram showing the energy levels where we have rotational states, and they're labeled here with the rotational quantum number, and the nuclear spin state that the molecule must necessarily be in if they're in this rotational state. And what we see here is that, oh, here we go. This is a very large number, this energy separation here. 170 Kelvin is enormous on an NMR interaction energy scale. So what we can do, I think I have a nice animation here, we can reduce the temperature of the hydrogen gas down to on the order of about 25 Kelvin. And we have 100% para enrichment, because what happens is the population falls down to this lowest rotational energy level. And that means that all of the nuclear spins must be in the spin singlet state. Okay, so I've shown these two cases. I think the state of this field is that many molecules can be polarized with parahydrogen, but there are only a few examples of molecules that can be polarized with parahydrogen to a high enough polarization level, or let's say more than a few percent, and purified from the reaction solution so that they could be used in a biological setting. And I think so pyruvate and fumarate are currently at that stage. And I have some numbers here for the current state of the art. So this paper came out from Stefan Glogler's group earlier this summer, where they achieve 8% carbon-13 polarization on pyruvate, and the solutions at 12 millimolar pyruvate concentration. Just give you a sense of the numbers that we're talking about here. And with fumarate, the current state of the art is about 18% carbon polarization at concentrations of about 100 millimolar. This is probably much higher than much higher concentration than would be necessary for in vivo metabolic imaging. You probably totally saturate the enzymes with the concentrations like that. It's probably a bit excessive. And here I put the numbers into a table to give a comparison there between dissolution DNP and FIP. So with pyruvate, DNP, you can achieve much higher polarization levels. I have some references down here for where these numbers come from. You can achieve polarization levels up to about 5 times higher than you can do with FIP. And with fumarate, the polarization levels are actually very similar. But an important difference is that with FIP, you need to use singly labeled fumarate, whereas with DNP, you can use doubly labeled fumarate. So the signals would in principle be twice as large because you have double the concentration of hyperpolarized nuclear spins. I can, oh yeah, perfect, that's shown here, nice. So with fumarate, we have it's a symmetric molecule, and we can have a carbon-13 on both sides or on just one side. And in these images here, this is a paper that's currently submitted work from Envision in Ulm in collaboration with TU Munich, where they compare FIP and dissolution DNP for in vivo imaging using hyperpolarized fumarate. And what we see is that essentially in these DNP images, the signals are about twice the intensity. But in the FIP case, although the signals are only half the intensity, it's still enough to take these metabolic maps showing malate to fumarate ratio and to extract information from the mouse in this case. Okay, so now that we're at that sort of point, where we can produce these molecules, fumarate and pyruvate with FIP, we have to ask about how are they actually being used? How are we learning about how these molecules can be applied? So in vivo experiments in humans and mice are intrinsically quite slow, and they introduce these ethical concerns where you have to get permission to carry out these experiments. And another method would be doing experiments in cell suspensions, but this doesn't necessarily mimic, mammal physiology particularly well. So what we're working on in our group here is lab-on-a-chip devices where we have 2D or 3D cell cultures in microfluidic devices, which in principle are able to mimic in vivo physiology much better. And you gain a lot of experimental control when you have a system like this, much more than if you're using a glass tube full of cells. So I show that here. So this is microfluidics, where we have, this is just an example of a microfluidic platform where you can introduce a sample and do many different types of things to it. Many different experimental steps can be implemented in just one device. But we have a problem when we use microfluidic devices, in that when we're using normal NMR samples, the sample volumes are of the order of hundreds of microliters. But if we go to a micro scale, the volumes that we have are much smaller. So the corresponding concentration limits of detection go up. So here I just give some reference numbers saying that in an NMR tube, you could expect to detect concentrations of species at about, you could detect species at about 10 micromolar concentration in a high field magnet with a cryoprobe, a sort of state-of-the-art equipment. Whereas for this microfluidic device with a 1 microliter sample, that number goes a lot higher, and you're sort of lucky to have millimolar sensitivities. So we say the problem is low sensitivity, and the solution, of course, is hyperpolarization. So what's shown here is the link between hyperpolarized NMR and microfluidics. So it's a two-way exchange, and I can run through this. So we can say that microfluidics supports research into hyperpolarized NMR. I mean, these two fields naturally seem to go quite well together, because microfluidics, you're concerned with getting a high degree of experimental control over a very small sample. And in hyperpolarized NMR, you're enhancing NMR signals from small samples. But exactly how that works, we say, well, in microfluidics, you've got a number of experimental parameters like short transport path lengths that mean you have reduced relaxation losses. You can integrate many steps onto one platform, so you have high experimental control. The small size scale is really important for many hyperpolarization techniques. So if you're wanting to develop KidNip, for example, where you need optical penetration into the sample, then working at the small sizes of microfluidic samples is highly beneficial. And it's very reproducible. You can repeat experiments quite easily, flowing samples through a chip, varying some parameter and acquiring spectra. And on the flip side of that, hyperpolarized NMR enables applications of microfluidics. And that is all because of improved sensitivity. So there are a number of applications like probing biological systems non-invasively, or quantitative analysis of chemical systems that you can do if you have hyperpolarization. And here is a diagram that puts a lot of microfluidic NMR experiments into context. So along this axis, we have the volume that's detected in the microfluidic experiment. So that's these vertical lines. And along here, we have the concentration limit of detection. For now, we'll ignore this mass limit of detection and we'll just look at concentrations. So these are the diagonal lines that go across. So we have this region here where we say metabolomics feasible in this orange region. And that corresponds to about 5 millimolar concentration limit of detection. Well, it's a bit arbitrary, but you probably need to be able to detect a few millimolar at least in a few seconds to be able to do metabolomics on a sample. And so many microfluid experiments hover around that range, but these experiments highlighted in red are hyperpolarized experiments, and that's where you can really push down to higher concentration limits of detection, or lower actually. Okay, and here I show a really nice example of that. That's this, where is it? Or perhaps it's not on here. This is a dissolution DNP experiment where a sample of pyruvate is polarized and injected into a microfluidic chip. That's shown here. And it flows through this microcoil with a cell suspension, and the conversion of pyruvate into lactate is observed spectroscopically. And so I think this is a really nice example that came out of Keivan Kashari's lab, showing that combination of hyperpolarized NMR and microfluidics. So we've highlighted the benefits of these lab-on-a-chip platforms, in that they mimic human physiology well. You have a high degree of control over what you're doing. And on top of that, you have a high experiment turnover rate. You can run experiments quickly and at quite low cost. These sort of platforms are just plastic chips that are cheap to produce. So we see quite a lot of parallels with the benefits offered by FIP, microfluidics. And so that's what we're working on now in Irene Marco's lab at IBEC in Barcelona. So we have these lab-on-a-chip platforms, and what we're doing is we're polarizing fumarate and pyruvate with FIP, and the idea is to inject those into these devices. So it's set up sort of like this, where you can have like a microcoil measuring a sample. You have cell media flowing through to keep your cells alive, and you can inject hyperpolarized fluid. And the goal, this is not data from microfluidic devices, this is coming soon. The goal is to observe that metabolism from pyruvate to lactate and use that to extract information about these biological samples as ways of testing new drugs, or to see how cells are responding to different stimuli. And with that, I'd like to finish and say thank you for your attention, and thanks to the whole MITMED team here at IBEC, and especially at Irene Marco, who is here leading the group, and for all the work that everybody has done. So thanks very much.